A single, fossilized tailbone rests on a foam pad inside a collections drawer. A deep, U-shaped gouge cuts directly through the dark stone of its lateral wing. This bone belonged to an Allosaurus, pulled from the dense sandstone of the Morrison Formation, an arid floodplain dating back about 150 million years to the late Jurassic. A cast of a Stegosaurus tail spike fits perfectly into this hole. But geometry alone does not prove a fight. A passing herbivore could easily step on the rotting carcass of a predator. The evidence shifts only when high-resolution micro-CT scans reveal a bulbous rim of bone remodeling around the puncture. It is a living callus. The predator survived the strike long enough to heal, leaving hard proof of active combat. How do we measure the physical limits of animals that no longer exist? Late Jurassic, about 150 million years ago. By this time, the supercontinent of Pangaea was well into its slow, grinding fragmentation. The descendants of the dinosaurs that survived the Triassic extinction had not just repopulated the continent. They had diversified into an array of forms that pushed the biological limits of terrestrial life. But to understand the true scale of their dominance, we have to physically pull the evidence out of the earth. It is a process that relies on brute force just as much as delicate precision. When a field crew uncovers a bone of this magnitude, in this case, the femur of a massive sauropod, the extraction process dictates everything that follows. We cannot simply lift the fossil out of the dirt. Millions of years of geological pressure have fractured the bone into thousands of pieces, held together only by the surrounding rock matrix. As the awl and brushes expose the surface, paleontologists immediately paint the bone with consolidant. This thin, penetrating glue seeps into the microscopic cracks. As the solvent evaporates, it binds the fragile fossil structure together. Once the top and sides of the bone are stabilized, the crew begins to dig a trench around it. This creates a rock pedestal that supports the fossil from below. Here, paleontology becomes an exercise in heavy logistics. To remove the bone without shattering it, the crew wraps the entire block in strips of burlap soaked in industrial plaster. As the plaster undergoes a chemical reaction and cures into a rigid shell, field workers undercut the pedestal, carefully flipping the encased fossil over to seal the bottom. The resulting plaster jacket for a single sauropod leg bone can easily weigh over 1,000 pounds. Moving it requires winches, steel sleds, and heavy machinery. The sheer physical labor required to extract these animals is our first direct, tangible measurement of their success. They were biological engineering projects of unprecedented scale. But as we map the quarry grid and record the precise location of every plastered bone, we confront a fundamental constraint in the science of deep time. The bone bed before us is a chaotic jumble of ribs, vertebrae, and limb bones belonging to dozens of different animals. It is tempting to look at this crowded mass of fossils and imagine a single, catastrophic event. We picture a sudden flash flood or a mudslide that buried a living herd in one afternoon. To test that assumption, we have to look away from the bones and read the stratigraphy. We examine the vertical column of sedimentary rock layers exposed in the quarry wall. The rock immediately surrounding the fossils is a coarse, cross-bedded sandstone, indicating the presence of a flowing river channel. But interspersed within and above this sandstone are layers of fine-grained mudstone, deposited by slow-moving water spreading across a floodplain. The bones themselves often show signs of weathering. They look as if they lay exposed to the sun and scavengers for months or years before finally being buried. This geological context tells us we are looking at a phenomenon called time averaging. The science of taphonomy, the study of how organisms decay and fossilize, shows that river bends and floodplain depressions act as natural traps. Over the course of decades, centuries, or even millennia, seasonal floods swept the carcasses of animals that died miles apart into the same geographic depression. As the bones settled into the sediment, time was compressed. A predator and a herbivore buried side by side in this quarry might have lived and died hundreds of years apart. We cannot confidently reconstruct a single afternoon in the late Jurassic from a time-averaged bone bed. But we can reconstruct the broader ecological patterns of the world they inhabited. When we analyze the chemical isotopes preserved in the Paleosols, the fossilized soils of the Morrison Formation, the romanticized image of the Jurassic period begins to evaporate. Pop culture often depicts this era as a continuous, lush, tropical swamp, but the oxygen and carbon isotopes locked in the ancient soil nodules tell a story of severe environmental stress. The data suggests a highly seasonal, semi-arid climate, It was characterized by brief, intense wet seasons followed by prolonged, punishing droughts. The rivers that deposited these sandstones frequently shrank to isolated, stagnant pools. This forced massive herds of animals to migrate across vast, dry floodplains in search of water. This harsh environmental reality brings us to a mathematical problem. We can examine the fossilized leaves and pollen grains preserved alongside the dinosaur bones in these sediment layers. Mapping the base of the food web. The flora of the late Jurassic was dominated by gymnosperms, plants that reproduce using exposed seeds, such as conifers, cycads, ginkgos, and ferns. The rock record provides direct evidence of a glaring absence. There are no angiosperms. There is zero trace of the flowering plants, broad-leafed trees, fruits, or fast-growing grasses that fuel modern mammalian herbivores. This presents a severe caloric constraint. Gymnosperm foliage is exceptionally tough, high in fiber, and difficult to digest. It offers a very low density of readily available calories. Yet, the sauropod dinosaurs whose bones we are wrapping in plaster jackets grew to weights of 30, 40, or even 70 tons. By modern biological standards, the energy required to simply move a body of that mass, let alone grow it, should be impossible to sustain on a diet of conifer needles and tough fern fronds. The environment was dry and seasonal. The food was poor in nutrients. And still, the animals grew to dimensions that defy the intuitive limits of biology. The solution to this paradox cannot be found by simply weighing the fossilized bone in the field. To understand how these giants actually functioned without collapsing under their own weight or starving to death, we have to transport these heavy plaster jackets from the dirt of the American West into the sterile, high-tech environment of a modern laboratory. We have to look inside the stone itself. The hum of a medical CT scanner fills a brightly lit hospital room. Long after the medical staff has gone home for the night, resting on the motorized bed, carefully wedged between foam blocks, is a fossilized cervical vertebra. It is a single neck bone from a late Jurassic sauropod, Brachiosaurus altithorax. In the field, this one bone required 3 people to lift it. It is massive, heavy, and seemingly solid, a testament to the immense physical load it had to bear. But paleontology has moved beyond merely measuring the external dimensions of fossils. To understand how an animal of this scale could actually exist in a world with low-calorie vegetation, we have to treat the skeleton not as a solid mass, but as an architectural structure. The heavy stone slides into the circular gantry of the scanner. Invisible X-rays slice through the fossil, mapping the density of the mineralized bone against the rock matrix that now fills it. On the technician's monitor, a cross-sectional image appears. Rendering line by line, it builds a 3-dimensional model of the bone's interior. What appears on the screen forces a complete recalculation of everything we know about dinosaur gigantism. The vertebra is not solid bone. The CT slices reveal deep, complex cavities called pleurocols boring into the center of the structure. Inside, the bone is mostly empty space. It is supported by a delicate, intricate network of thin bony struts and reinforcing walls, resembling the internal framework of an airplane wing. SRC03, Weedle, 2003. This direct structural evidence completely reframes our understanding of the animal. If we calculate the volume of a sauropod's neck assuming it was made of solid bone, the resulting mass is catastrophic. The animal would barely be able to lift its head. The sheer metabolic cost of dragging that weight across a semi-arid floodplain would be unsustainable. But the CT scan proves that math was wrong. The extensive pleurocols drastically reduced the specific gravity of the sauropod skeleton. They were building maximum length and height with a fraction of the raw material. This internal architecture provides more than just a reduction in weight. We can apply the principles of comparative anatomy, looking for modern animals with similar skeletal structures. When we do, we find an exact match in only one living group: birds. Modern birds possess a highly specialized respiratory system featuring air sacs. These are balloon-like extensions of the lungs that spread throughout the body cavity, even penetrating the hollow cores of their bones. This system allows birds to extract oxygen during both inhalation and exhalation. It creates a continuous, unidirectional flow of fresh air that is drastically more efficient than the mammalian diaphragm. Finding these deep pneumatic cavities inside the fossilized vertebrae provides strong inference that sauropods possessed a similar avian-style respiratory system. SRC03, Weedle, 2003. This was the evolutionary cheat code that allowed dinosaurs to dominate the ecological vacuum left by the Triassic extinction. It solved two massive problems simultaneously. First, it lightened the skeleton enough to allow for extreme size. Second, the hyper-efficient oxygen exchange helped power the immense physiological demands of a body weighing 30 to 70 tons. Even in a harsh environment. With the physical constraints of weight and respiration mitigated by this internal engineering, sauropods were free to push their feeding strategies to the extreme. The fossil evidence shows that different lineages adapted their immense, lightweight frames to harvest the tough gymnosperm flora in entirely different ways. We can examine the articulated skeleton of Brachiosaurus. The extreme elongation of the forelimbs relative to the hind limbs, combined with the upward angle of the neck base, points to an animal specialized for high browsing. It acted as a biological crane. Utilizing its lightweight, air-filled neck, it harvested the upper canopy of conifer forests, accessing a vertical food source that no other herbivore in the ecosystem could reach. We see a different physical reality when we reconstruct another common Morrison giant, Diplodocus. The structure of its cervical vertebrae heavily restricted vertical movement. It could not lift its head high into the canopy. Instead, its neck functioned like a massive, horizontal boom extending from the shoulders, counterbalanced by an incredibly long, whip-like tail. This geometry allowed Diplodocus to stand in one place and sweep its head in a wide lateral arc. It could vacuum up vast quantities of low-lying ferns and sea cycads with minimal energetic cost. However, a strict constraint limits how far we can take these reconstructions. We can calculate the mechanical advantage of the joints, and we can measure the hollow spaces in the bone using a CT scanner. But we cannot know the exact mass or behavior of the soft tissue that once surrounded it. We do not know exactly how much food a Diplodocus had to consume in a single day. Nor do we know exactly how efficiently its digestive tract processed the fibrous vegetation. We can only measure the architectural limits of the scaffolding left behind. But that scaffolding alone tells us that sauropods were not sluggish, overgrown reptiles doomed by their own size. As early paleontology often assumed, they were highly optimized, lightweight biomechanical engines perfectly adapted to extract energy from a poor landscape. This level of physical optimization in the herbivores creates an undeniable evolutionary pressure. If the prey animals have engineered themselves into towering, 30-ton fortresses of bone and muscle, the predators that share their environment cannot rely on brute force alone to bring them down. To understand how the carnivores of the late Jurassic adapted to this arms race, we have to take another skull from the Morrison Formation, place it back into the digital realm, and subject it to forces that would shatter a lesser structure. On a computer monitor in a biomechanics lab, a wireframe mesh slowly rotates. It is the skull of Allosaurus fragilis. When we pull a skull like this from the field, it is an intimidating object. It is defined by a deep snout, small bony crests positioned just in front of the eyes, and jaws lined with serrated, backward curving teeth. For decades, this visual impression dictated how we assumed the animal hunted. Because it was the apex predator of the Morrison Formation, the natural assumption was that it operated much like a modern lion or a crocodile. We pictured it using massive jaw muscles to bite down, grapple, and crush its prey through sheer bilateral force. But visual intuition is not a reliable metric in deep time. To truly understand the mechanics of this predator, we have to strip away our assumptions about modern mammalian carnivores. We must test the fossil as an architectural unit. We do this using a method called finite element analysis, originally developed by civil engineers. This software tests how stress distributes across the steel beams of a bridge or the fuselage of an airplane before it is ever built. Now, it allows paleontologists to apply simulated physical loads to digital models of fossil bones. The digital mesh on the screen is divided into hundreds of thousands of tiny, interconnected geometric blocks. These are the finite elements. By assigning these blocks the known material properties of bone, we can anchor the back of the digital skull exactly where the neck muscles would attach. Then, we apply a simulated force to the teeth. The paleontologist running the simulation applies a strong lateral force to the model. This simulates the stress of a predator biting down and violently shaking its head side to side, or struggling to hold down a thrashing, multi-ton sauropod. Instantly, the heat map on the screen flares red and yellow around the jaw hinge and the thin struts of the skull roof. The red indicates areas of critically high strain. The strong inference drawn from this data is that the Allosaurus skull was remarkably weak against lateral twisting forces. If it hunted by grappling and wrestling massive prey to the ground, its own skull would likely fracture under the stress. SRC01, Rayfield. 2001. This seems to present a paradox. How could the apex predator of the late Jurassic have a weak skull? The answer requires changing the angle of the applied force. The paleontologist resets the simulation and applies a different load. This time, the force is directed straight up through the teeth. This simulates a rapid, vertical impact. As the digital force hits the upper jaw, the heat map remains largely blue and green, indicating very low stress. The architectural design of the skull features multiple large fenestrae, the empty windows in the bone that reduce weight. The simulation reveals that these empty spaces act as a highly efficient shock absorber. The force of the impact is channeled away from the teeth and smoothly dissipated along the reinforced struts of the snout and the skull roof. The structural model demonstrates that the Allosaurus skull exhibits a safety factor optimized to withstand vertical impacts rather than bilateral struggling. SRC01, Rayfield. 2001. This revelation led to the proposal of a radically different hunting strategy, often referred to as the hatchet bite hypothesis. Instead of biting and holding, this speculative reconstruction suggests a completely different motion. Allosaurus may have used its strong neck muscles to drive its open upper jaw downward into prey like an axe. It could use its serrated teeth to slash deep, bleeding wounds before rapidly retreating to avoid the thrashing weight of a 30-ton herbivore. It is crucial, however, to recognize the constraints of this digital method. Finite element analysis measures the structural limits of the bone. It does not film the animal's behavior. Because of this gap between structure and action, the hatchet bite hypothesis remains a subject of active debate within the scientific community. Other researchers point out that the simulation relies on necessary assumptions. The computer model has to estimate the exact thickness of the missing cartilage and the precise volume of the jaw muscles. A minority view argues that the skull's ability to absorb vertical impact does not necessarily prove an axe-like strike. It might simply mean Allosaurus had a very lightweight skull adapted for quickly stripping flesh from carcasses, or that it possessed a highly varied feeding style that never required massive bite forces. What the digital method does prove, beyond debate, is that Allosaurus was not a blunt instrument. It was a precision-engineered system, operating within strict physical limits to survive in an ecosystem dominated by giants. But a computer model, no matter how sophisticated, remains an abstraction. It is a simulation of what could happen, a map of potential forces. To prove that these massive predators actually engaged in physical, life-and-death combat with the herbivores of the Morrison Formation, we need evidence that cannot be debated as an artifact of a software parameter. We need an artifact of violence that was caught in the act, recorded in the bone itself, and preserved in the stone. Resting on a foam pad in a collections drawer is a single, isolated tail vertebra. It belongs to an adult Allosaurus fragilis. At first glance, it is an unremarkable piece of dark, fossilized bone recovered from the dense sandstone of the Morrison Formation. But when you rotate the bone under a direct light, a catastrophic structural failure becomes visible. Gouged deeply into one of the lateral processes, the bony wings that extend from the side of the vertebra, is a massive, U-shaped puncture wound. This is not a clean break. It is not a typical post-mortem fracture caused by the crushing pressure of overlying rock. The bone has been violently penetrated. To understand exactly what caused this damage, paleontologists must approach the fossil not merely as a biological specimen, but as a forensic crime scene frozen in deep time. In the same laboratory, a researcher retrieves a heavy, conical cast. It is a thagomizer, the bone spike that tipped the tail of Stegosaurus stenops, the heavily armored herbivore that shared the Morrison floodplains with Allosaurus. For decades, the function of these Stegosaurus tail spikes was debated. Were they purely for display, or were they functional weapons capable of penetrating the thick hide and muscle of a predator? The researcher takes the cast of the Stegosaurus spike and slowly lowers the tapered point toward the puncture in the Allosaurus vertebra. The fit is perfect. The U-shaped puncture wound in the Allosaurus caudal vertebra precisely matches the geometry of the Stegosaurus tail spike. SRC02, Carpenter et al. 2005. This physical alignment provides direct geometric evidence of an interaction. But geometry alone does not tell the whole story. The critical constraint in paleoforensics is taphonomy, the timeline of decay. If an Allosaurus died of old age or starvation, its carcass would lie rotting on the floodplain. A passing Stegosaurus might have inadvertently stepped on the remains, driving a spike through the vertebra of a long-dead predator. To prove this was an act of combat, we need evidence that the bone was alive when the strike occurred. We place the punctured vertebra into a micro-CT scanner. This generates high-resolution cross-sections of the bone tissue directly surrounding the wound. The digital slices reveal something extraordinary. The margins of the U-shaped hole are not sharp, jagged, or brittle. The edges are blunted. The internal matrix of the bone shows distinct, bulbous growth patterns around the injury. This is a callus. It is the undeniable signature of bone remodeling, the physiological process of a living animal attempting to heal a traumatic fracture. This pathology provides direct evidence that the Allosaurus not only sustained the strike from the Stegosaurus spike, but that it survived the injury long enough for the bone to begin healing. SRCO2, Carpenter et al. 2005. This represents a rare, macro reframe in paleontology. We are no longer relying on the mathematical probabilities of a computer simulation. We have hard, physical proof of active, life and death combat. The Stegosaurus did not just wear its tail spikes for display. It possessed the biomechanical torque required to drive solid bone through the dense muscle and skeletal structure of an attacking apex predator. This interaction also provides strong inference regarding the rest of the Stegosaurus anatomy. The massive, alternating plates that run along the animal's back have long captured the public imagination. But when we examine the cross-section of a Stegosaurus plate under a microscope, we find that the bone is incredibly porous. It is riddled with wide vascular channels that once held a dense network of blood vessels. While speculative reconstructions often suggest these plates could flush with color for display, the structural reality presents a severe constraint. The plates are highly vascularized and relatively thin. This renders them far too fragile to serve as primary physical armor against the impact-absorbent jaws of an Allosaurus. If an apex predator bit down on one of these plates, the bone would shatter, and the resulting blood loss could be fatal. Therefore, the plates likely served functions tied to thermoregulation or display. The heavy lifting of defense was relegated entirely to the lethal physics of the tail spikes. We have now measured the immense scale, the internal engineering, and the violent combat of the giants that dominated the late Jurassic daylight. But the Morrison Formation, with its coarse sandstones and abrasive floodplains, has a massive blind spot. It is a geology that favors the heavy and the robust. It obliterates the delicate, the small, and the soft. To understand the full scope of the biological vacuum that the dinosaurs filled, we must leave the arid American West. We must travel to an environment where the geology itself was perfectly engineered to arrest decay. Late Jurassic, about 145 million years ago. A map shift takes us thousands of miles away from the brutal, seasonal floodplains of North America. We arrive in what is now the Bavarian region of Germany. In the late Jurassic, Europe was not a continuous landmass. It was a warm, shallow archipelago, a sprawling network of tropical islands surrounded by calm, sunlit seas. In a quiet museum hall, a raking light is angled low across a rectangular slab of incredibly fine-grained, pale limestone. The light catches faint, unmistakable impressions radiating outward from the fossilized bones embedded in the rock. The fossil is Archaeopteryx lithographica. The impressions perfectly preserved in the stone are asymmetrical flight feathers. This fossil forces us to confront a severe taphonomic constraint. Why do we find feathers preserved down to the microscopic barbules here, while the massive bone beds of the Morrison Formation yield almost nothing but stripped, bare skeletons? The answer lies in the specific chemistry of the Solnhofen limestone. During the late Jurassic, this region was defined by isolated, hypersaline coastal lagoons. These shallow bodies of water were periodically cut off from the open ocean. Under the intense tropical sun, the water evaporated rapidly. This left behind a brine so salty and toxic that very little could survive in it. More importantly, the water column became stratified. The warm, oxygen-rich water stayed near the surface, while the dense, hypersaline brine sank to the bottom. This created a completely anoxic, or oxygen-free, death zone on the lagoon floor. When an animal like Archaeopteryx died and sank to the bottom of this lagoon, It entered an environment perfectly engineered to arrest decay. There were no scavenging crabs, no bottom-feeding fish, and most crucially, very few of the oxygen-dependent bacteria that normally dismantle soft tissue. Blanketed rapidly by a fine rain of carbonate mud, the animal was sealed in a chemical vault. The Solnhofen limestone does not just preserve bones. It preserves the shadows of biology that are usually erased by time. The feathers of Archaeopteryx provide a direct morphological correlate to aerodynamic capability. The feathers on its wings are asymmetrical. The leading edge of the feather is narrow and stiff, while the trailing edge is wide and flexible. This is strong inference for aerodynamic function. This exact geometry is required to generate lift and prevent the feather from twisting under the pressure of the air. Yet, looking closely at the slab, the animal is a paradox. Alongside the aerodynamic feathers, the direct evidence shows a jaw lined with sharp, conical reptilian teeth. 3 distinct fingers tipped with functional claws extend from the wing. A long, bony tail made of individual, articulated vertebrae trails behind the body. Archaeopteryx sits at the precise intersection of deep evolutionary transition. It carries the heavy structural legacy of its theropod dinosaur ancestors directly into the sky. However, the presence of aerodynamic feathers introduces an active debate that highlights the limits of our reconstruction methods. The exact mechanism of how this animal launched itself into the air remains contested. Did flight evolve from the trees down, with animals using their feathers to glide from the canopy to the ground? Or did it evolve from the ground up, with bipedal runners using their feathered arms to generate lift while chasing prey or scaling steep inclines? Because the Solnhofen limestone preserves the dead animal flat on a slab, we can measure the aerodynamics of the wing, but the launch sequence is highly model dependent. The origin of flight remains a boundary we cannot completely cross with static fossils alone. What we do know is that when the early avialians took to the air, they did not find an empty sky. The Solnhofen limestone also preserves the remains of pterosaurs, flying reptiles that had conquered the air millions of years earlier. Unlike the feathered wings of Archaeopteryx, pterosaurs utilized a wing membrane stretched over a single, enormously elongated fourth finger. The sky was fully partitioned, engineered by 2 entirely different biological solutions to the problem of gravity. The daylight hours of the late Jurassic were entirely dominated. Sauropods engineered their bodies to consume the forests. Theropods engineered their skulls to hunt the giants. Pterosaurs and early avialians engineered the sky. Every major ecological niche on the terrestrial board was locked down by the descendants of the Triassic survivors. But as the sun set over the gymnosperm forests, the temperature dropped, and the giant reptiles slowed. In the darkness beneath the massive roots of the sea cycads and ferns, the final piece of the Jurassic ecosystem began to move. It was an empire built not on size or raw physical power, but on the desperate need to remain unseen. To find the evidence of its existence, we must abandon the scale of the dinosaur completely. We must learn to hunt for fossils the size of a grain of rice. Middle Jurassic, about 164 million years ago. A map shift brings us to the Daohugu beds in northeastern China. In the preparation lab, a technician sits motionless before a high-powered microscope. In her hand is an air scribe. It is a pneumatic tool tipped with a microscopic tungsten carbide needle that vibrates thousands of times per minute. She is not clearing rock from a femur the size of a tree trunk. She is carefully vibrating away grains of matrix from a tooth that measures exactly 3 millimeters across. For decades, this tiny scale was the absolute limit of our knowledge regarding Jurassic mammals. The narrative generated by this extreme scarcity of evidence was simple. Mammals supposedly spent the entire reign of the dinosaurs as terrified, uniform little shrews. They were cast as the ultimate ecological victims, trapped in a nocturnal bottleneck, waiting in the dark for a meteorite to clear the board. But this assumption was a byproduct of sampling bias. Finding a 3 mm tooth requires screen-washing tons of sediment through fine mesh. It is a needle in a geological haystack. We assumed early mammals were all terrestrial shrews because they're delicate. Specialized skeletons were simply too fragile to survive the abrasive river systems that preserved the giant dinosaurs. Then, the Daohugu beds yielded a slab of fine-grained volcanic ash and lake sediment. When the rock was split, it did not reveal a tiny terrestrial shrew. It revealed Castoricauda lutrasimilis. This animal shattered the myth of mammalian uniformity. Under the microscope, The Castoricauda fossil preserves direct evidence that forces a massive reframe. The rock surrounding the skeleton is stained with a dark carbon halo. When magnified, this halo resolves into the distinct, individual filaments of dense underfur and longer guard hairs. This is the earliest definitive proof of a mammalian pelt. But the true revelation lies in the architecture of the skeleton itself. The tail vertebrae are broad and flattened, exactly mirroring the skeletal structure of a modern beaver. Furthermore, the soft tissue preservation reveals distinct webbing between the toes of the hind feet. The 3 mm teeth are sharply recurved and interlocking. This is the specialized dental signature of an animal that catches and eats slippery fish. SRC04, G. al. 2006. Castoricauda was a semi-aquatic, swimming mammalia form, roughly the size of a modern muskrat. It was diving into lakes and rivers, hunting fish right under the feet of the dinosaurs in the Middle Jurassic. The mammals were not just hiding, they were actively exploring complex ecological niches. SRC04, G. al. 2006. Yet, the true legacy of the Jurassic mammals is not found in their ecological diversity. It is found in the internal engineering forced upon them by the darkness. Because dinosaurs heavily dominated the daylight, mammals were restricted to the night. This nocturnal bottleneck imposed a severe evolutionary constraint. In the dark, the acute color vision favored by reptiles is useless. Survival depends on the processing of faint sounds and complex odors. When we put the tiny, fragile skulls of these early mammals into a high-resolution micro-CT scanner, we can trace a profound anatomical migration. In the early ancestors of mammals, the jaw hinged upon 2 small bones at the back of the skull. But as these animals adapted to the dark, the CT slices show these bones physically shrinking. They detach from the jaw hinge entirely and migrate upward into the skull. They become 2 of the 3 tiny bones of the mammalian middle ear. By sacrificing a complex jaw hinge, these animals gained an unprecedented sensitivity to high-frequency airborne sound. They could hear the faint rustle of an insect in the leaf litter, or the quiet footfall of a predator in the pitch black. To process this massive influx of new auditory and olfactory data, the mammalian brain had to physically expand. The neocortex, the region of the brain responsible for sensory perception and complex cognition, began to wire itself in the shadows of the Jurassic gymnosperm forests. We are looking at the foundational blueprint of our own neuroanatomy, the brain that allows a human being to program a CT scanner, to run finite element analysis on a bone, and to comprehend the vastness of deep time, was forged by the evolutionary pressures of avoiding dinosaurs in the dark. As we look back across the evidence, the true nature of this world comes into focus. It was not a landscape of sluggish, overgrown monsters waiting for extinction. From the moment the Central Atlantic magmatic province volcanism cleared the Triassic board, life pushed the boundaries of terrestrial physics. Sauropods engineered their bones with air to achieve impossible size. Theropods engineered their skulls to absorb catastrophic impacts. And early mammals engineered their senses to conquer the night. We cannot hear the heavy, unidirectional breathing of a Brachiosaurus moving through the arid Morrison floodplains. We cannot watch the exact choreography of an Allosaurus suffering a strike from a Stegosaurus tail. The sounds, the colors, and the behaviors of this world are forever trapped behind the boundary of deep time. What survives is the scaffolding, the stone and the bone. But by treating that scaffolding not just as rock, but as an architectural structure that we can measure, stress test, and scan, the silence of the fossil record breaks. The physical limits of the bone tell us exactly what these animals were capable of, and how completely they mastered the machine of the earth.